in total 4 lectures. So the first 3 will be devoted to chromatin, and the last one to cancer. So that's the plan. So first 3 lectures on chromatin, what I call chromatin physics. And then 1 lecture on cancer evolution. Almost unrelated to the first 3. Okay, so basically the theme of this lectures on chromatin is basically to look at the genome as a physical object. And that's the underlying theme. And I imagine you have very different backgrounds, so I will try to get you on board of general genomics, talk about some genomic techniques, experimental techniques, not right now, later on. In general, talk about genomes of different organisms. So the first thing that's useful is to keep in mind that different organisms are vastly different in terms of their genetic and genomic organization. So I'll be switching between organisms. I'll be mostly talking about mammalian systems and higher eukaryotes in general. But I will be talking about bacteria and single-cell eukaryotes. So let's for human, mouse will be one system. Then in the other extreme, we'll be talking about bacteria. And somewhere in the middle are single-cell eukaryotes. We don't really know much about them, not as much as we know about human and bacteria historically. So single-cell euks. We do know a lot about yeast. But generally, there are simple early metazoans, early multicellular eukaryotes, and single-cell eukaryotes, and knowledge about them is kind of limited. So I'll be talking about yeast and implying that that kind of works for other simple eukaryotes. So the first thing about, so let's talk about genome. So how long is the genome? And then we'll talk about various other aspects of genome. So how long is a human genome? Anybody? So I want you to talk, sort of. I know you asked lots of questions, so I'll be asking you questions, and I want you to answer. The length of the human genome? 10 to the 9. Okay, that's the right order. Any prefactors? 3. Okay, so that's the length of the haploid human genome. So how many human genomes do we have in each cell? Somebody said 2, somebody said something else. So how many copies of the human genome do we have in each single nucleus? Or in each single cell, which is even more tricky question. The right answer, it depends on the cell. It's like, do you want tea with lemon or with milk? It depends on the tea. British jokes. Okay, good.
I disagree, actually.
You fight with the Brits. So depending on the cell. So first of all, it depends on the stage of the cell cycle. So in G1, so cell cycle, we'll need a cell cycle here. There is G1. Let me draw it different. There is G1. There is S. There is G2. There is M. And then there are various variants of this called G0. There are quiescent states. There are senescent. There are various many special states where a cell can go into from G1.
Sorry, so I mean, if you take a random cell in your body, it's probably not dividing. Right?
If I take a random cell in my body, it's not divided. If I take a random cell in my body, as we'll find out very soon, it's 50-50 is going to be bacteria. Not because I brought a bacterial infection from the airport yesterday. which I had good chances of. But because half of our cells are actual bacterial cells. But yes, if we take a random cell in my body, it's not dividing. Most of our cells are in some form of G1. But, okay, we'll come back to this in a second. So in G1, we have two copies. We have two copies. In G2, it's more interesting. We need to, okay, so what does it mean two copies? I have one maternal and one paternal. Right? You need to, it's not going to be incredibly important, but just kind of. Okay, G1 is a state of a cell. So a cell, what cell does, so it has its DNA. At some point it divides, it replicates, it needs to divide. To divide, it needs to replicate its DNA. It needs to copy its DNA. So to copy its DNA, it takes some time, and that's done at the S. I need another color. At the S phase. So that's where replication takes place. So the cell needs to make a copy of its DNA. In G1, a normal somatic cell has two copies of DNA, one from my mother and one from my father. In G1, what happens? It creates a copy of each, and I have now two maternal. So okay, let me try it more carefully. In G2, in the S phase, I replicate. In G2, I have two copies, and I have two sisters. And that's something that people typically confuse. So copies are called homologs. So I have chromosome 1. Can you see the board? I'll be using board space kind of this way. So for example, if I look at chromosome 1, I have each nucleus has chromosome, paternal chromosome 1 and maternal chromosome 1. Right? Chromosome 1 maternal, chromosome 1 paternal. Everybody is familiar with the signs. You know, when you teach to an audience that you don't know, it's kind of challenging because, like, when I say vector, a vector means a totally different thing for a biologist and a physicist, right? When I say matrix, it means very different things. So, so there are two copies of the chroma. During replication, you get an extra copy of each. During replication, I get a pink. It's pink, isn't it? I get a new chromosome 1 maternal, and I keep the old chromosome 1 maternal, and then I have a new chromosome 1 paternal, and a new chromosome 1, and an old chromosome 1 paternal. So total, I have 4 copies of chromosome 1. Chromosome 1. So each chromosome is present now in 4 copies. And these copies are not all 4 identical. These 2 are identical. Practically identical. These 2 are identical. They're called sisters. So these are 2 sisters, and these are the 2 sisters. So in G2, typically we have a prefactor of 4 in front of this. So these are prefactors. These are prefactors that go, it's either 4 or 2 that go here. So it can be 2, 4, something in this range. Yes?
How long are these phases?
I plan to talk about this. We'll come to the time scales at some point.
So the first of the G1, the word copies is in quotes because they're not.
Say it again.
The copies is used in 2 different senses, right? I mean, so it's not really.
They're not exactly copies, so they're not identical. Your paternal and your maternal are different.
Right. So what's
They're different. Okay. How different are we from each other? as 2 human individuals, roughly. In how many positions out of 3 to the 10 to the 9? 0.1%. Exactly, 1 in 1,000. 1 in 1,000 bases are different between paternal and maternal, assuming that a mother and a father are unrelated. So roughly 99.9% identical. These things are much more identical, practically exactly identical except for mutations, and mutation rate is very, very low. So less than 1 per chromosome, less than 1 mutation per genome. So no errors to the first approximation. Okay, so what's important are the ballpark. So numbers, 2 to 4. Some cells, however, have more copies. So if we take my random, if you say, so take my liver cell. In the liver is typically polyploidal. So it's in some sort of G1, but instead of 2 copies, it has 4, some people say 6 or 8 copies of the genome. The liver is typically polyploidal, and we don't really know much about ploidy in different body types, in different body organs, in different individuals. Kind of, it's something that people are exploring now. Okay, so that's the ballpark. So how long is a bacterial genome? How many base pairs? 110 to the order of 10 to the 6. Okay, so that's an order of a megabase. Okay, any prefactors? That's good. That's right. So a typical prefactor is somewhere of the order of 4 or 5. You can say 5, but we understand that that's an order of magnitude. So some bacteria are short, like Mycoplasma. Some bacteria are long, but that's not. Yeast is somewhere in the middle. Yeast is somewhere of the order of 10 to the 7 with some prefactor. Okay, so that's good. So how many, before we go any further, how many genes and how long are the genes? Let me erase some of this. Some of this stuff. So we'll come back to the stages of the cell cycle. How many genes are there? So human, how many genes do we have?
25,000.
Yeah. And bacteria, typically? No, a few thousand. And yeast is somewhere around 6,000, somewhere in thousands. So numbers are not that different. So the genomes are basically 3 orders of magnitude longer, but the number of genes is only a factor of basically 1 order of magnitude different. Yes?
Could you define what gene is?
People can fight forever what gene is. So operational definition of a gene is a protein coding gene. It's a piece of DNA that encodes a protein. And there might be variants, so you can splice it differently, you can dice it differently. So it's a piece of DNA that encodes a single protein. That's going to be my operational definition of a gene. How long are the genes? What's the length of each gene? More like 10 to the 4 in human. With some prefactor, maybe 2. I was looking at the literature today, kind of, there is no agreement on this, but say 10 to the 4. How long are bacterial genes? Typically 10 to the 4 base pairs. So what's the difference between bacterial and eukaryotic genes is that our genes consist of exons and introns. That's exons.
We're going to try to write a little bigger because you're
Okay.
recording and also the students.
Okay. Exon 1, then there is an intron, then there is another exon, then there is another intron. For some weird reason, and so on. for some weird reason, the coding information in the genes, in the human genes, is split into pieces. And there are long uncoding regions that are inserted in the middle of the gene for no obvious reason. And when the gene is being read as one continuous region, after it's being read, these pieces are spliced out, being cut out. And you form a final product that consists of fused together exons. Now, bacteria and simple eukaryotes don't have this. So that would be, so these are these exons.
So at what stage of the cell?
So at what stage of cell evolution introns, so there are some introns in yeast, but not many. And there is a long debate of whether introns invaded at some point, or whether it was an evolutionary advantage in getting introns. So somewhere at the stage of multicellular organisms, introns start to emerge. But not, somewhere at the invention of multicellularity. I don't know exactly when. So in bacteria, you don't have this mass. In bacteria, you have one continuous coding sequence. So it's one continuous coding sequence that encodes a protein. Yes?
Sorry, I think this is
Oh, excellent.
discovery of exons and then the removal of electrons.
So this is happening when the piece of DNA is being read. What do you produce? You produce RNA, right? So when you produce RNA, these pieces are being cut out. When exactly, it's not known. Now there are some evidence that this is happening as RNA is being produced. The classical textbook picture is that first RNA is fully produced, then it floats somewhere, and then somewhere in the splicing factory, these pieces are being cut out. It's not really known exactly when, but this is during transcription, during gene expression. Coding sequence. In bacteria, this is one continuous coding sequence. This is a gene. One gene, as we agreed, codes for one protein. Typical length of a protein is a few hundred amino acids. I assume you know this. So if each amino acid is encoded by 3 nucleotides, the typical length of the gene should be of the order of 1,000 bases. If this gene that produces a protein, typical length of a protein is, say, 300 amino acids. And then each is encoded by 3 base pairs, so this is about of the order of 1,000 base pairs. So one gene is approximately 1,000 base pairs. This in human is also of the order of 1,000 10 to the 3 base pairs. But because there are all these useless insertions, that makes genes 10 times longer. Yes?
Is it known the advantage of alternative splicing, so putting different exons together in this form?
This is something that's being actively used. So thank you, Armita. It's a great point that one advantage in having this architecture is that you can, instead of connecting them 1, 2, 3, I can cut out 2, and then I will have another gene which would consist of exons 1 and 3 together. Or I can just produce 1 and 2 and not produce exon 3, cut out exon 3. So as a result, there are lots of alternatives for every single gene in eukaryotes. There are alternatives, so this is called alternative splicing. There are also alternative starts. I may decide to start somewhere here, and that's going to be my number 1, and that's going to be my exon number 2.
What fraction of genes
I don't know the number. If you look at the genome browser, it looks like practically every gene has some either alternative starts, or alternative ends, or alternative exons. So it looks like, to the first approximation, I would say half of the genes use alternative splicing. And you can check on Google. So that would be my guesstimation. I would say every other gene use alternative splicing. At what conditions alternative splicing is being used, that's another question. But sort of at this point, so the question is, what are the numbers? So the length of a gene in eukaryotes and in bacteria. So coding part is this, but the length of the genes are much longer in eukaryotes than in bacteria. In yeast, they are of the order of 10 to the 3 bases, so they're more like bacterial ones. They might be somewhat longer, but maybe a prefactor here.
So in human, do you count the intron also as a part in the length of the gene?
Yes, exactly. So the length of the gene, this number includes, that's what I was about to draw, includes this. So genes are long. They collectively, so coding part collectively occupies, and we can estimate that. I say a typical length, let's put a prefactor here. I think in human I would safely put 2 or 3 here, but I don't know. I would put 2 10 to the 3 bases. What fraction of the genome using these numbers would be coding? Let's put numbers together. So I say 25,000 times 2 10 to the 3. So that's how much I expect coding would be. So this would give me 50 times 10 to the 6 out of 3 10 to the 9. I think I got even less than I expected, but that's okay. So this would give me 10 to the minus 2. And here I would have, okay, do it carefully. So this would give me 6, 7. I want percent. So this will be 10 to the minus 3 times 6. So this is, okay. Yeah, about 1%. Okay, fine. Okay, 12. Okay, 13. Okay, about 1 point something percent. So something like this. Okay, 15, 50, 60. Okay, whatever. 1.5. 1.5%. So that's about right. But the total length of the genes is 15%, if not more. If not, depending on this prefactor. Maybe 15%, 20% of the genome is covered by genes. So don't have this impression that these are tiny, tiny things. This is like comparable part of the genome. That will be important, actually. Yes?
So the introns are also replicated. It's just that they're not transcribed.
No, everything is replicated. I think what you meant to ask is introns are also transcribed, but not translated.
Oh, okay.
Okay, let's have a note about this. So this is DNA. The DNA is being copied. This is replication. Now, at the same time, genes with their introns, and everything is replicated. Genes are being transcribed, rewritten, right? Transcribed into RNA. So this is DNA. This is DNA. That's a DNA. That's a copy. We know that these are called sisters. So you produce RNA that includes all the exons and introns. This is transcription. And then from this you produce a protein which is totally different beast. So that's a protein. And this process is called translation. this. Okay, translation, transcription, replication. So let's keep it on the board. Okay, cool. So again, so just to go through this one more time. So we have DNA. DNA is being copied. So now we have 2 copies of DNA. That's replication. That's what we spoke about when we discussed the cell cycle. We'll come back to this. Each gene can be turned on or off. Gene is a part of the DNA that encodes for stuff. In my definition, codes for proteins. Now, genes are being transcribed. Another word for this is expressed. So genes can be expressed or transcribed. This means that the gene is being read by the cell. When a cell reads a gene, it reads its DNA sequence, including introns and exons, and produces an RNA molecule. That's the one. That is the exact copy of the one in DNA. And I'm saying exactly, I know there are some small, as always in biology, there are more exceptions than rules. So I'm giving you rules, but there are tons of exceptions. My brain is like firing, no, not quite. Yeah, there are exceptions. You have RNA produced from the gene. And then this RNA is now you have a process in which you cut out these things. Okay, that's what let me redraw it. Let me just add this cutting out. So then you cut out these things. This process of removing introns is called splicing. So that's this interesting process of cutting out unnecessary parts of the gene. And after splicing is complete, you get this magenta genetic information to produce a protein, and you produce a protein by the process called translation. So this way, information from DNA gives you a protein. And that's how genes are being transcribed. Now, yes, that's actually a great segue into what I want to talk about next.
Is this part of the cycle that you do?
This is happening all the time.
This is happening.
All the time. Except in the M phase. Genes are being transcribed in G1, in S, and in G2. But they're not being transcribed during M phase. Yes?
And this splicing step, this is the one we're not sure about, whether it happens before.
It may happen at the same time as transcription, called co-transcriptional splicing. That's what people start to believe now. A textbook would tell you that things are happening in steps. But yes.
It might be the case that you're just copying the coding regions.
No. You're copying everything. So you're certainly copying everything. But the question is, when do you cut things out? That may depend on a ton of different things.
Can you say what the time scale is?
Yes. So the time scales. Okay, the time scales, and that's something I want to talk about again in different organisms. It's quite different. So let's talk about time scales. So the whole cell cycle, how frequently do cells divide? So division time. Any guesses? How frequently do mammalian cells divide when they divide? How long does it take, roughly? What do you expect? It's not kind of, that's an unusual case. Typically once a day. if you have cultured cells, the division time is roughly of the order of 1 day, let's say 24 hours. The extreme, and I'll talk about extremes in a second.
That's the full cycle?
That's the full cycle. That's the full cycle. Most of the cells that don't divide, but those that divide roughly divide in the petri dish, in the lab. They divide once a day. And I believe normal cells that regenerate stuff divide at about the same rate. And those that produce blood, I should know these numbers, but I don't remember. But different parts of the blood kind of, you have, among dividing cells, you need to generate new epithelial cells, you need to generate new blood, right? Their rates of divisions are quite different, but generally that's a ballpark. Bacteria, how fast do bacteria divide?
41 hours.
Yeah, of the order of an hour, let's say, 1 hour. Yeast, comparable to that. Now, extreme cases are quite interesting, and so in animals, for example, during animal development, cells can divide extremely fast. The number that shocked me recently was that zebrafish, these cells divide every 15 minutes during very early stage of embryogenesis. So this is like extremely, extremely fast. So in the limit, this can be 15 minutes. In bacteria, again, sort of the limit would be also of the order of maybe 20 minutes, as far as I know. Now, different stages of the cell cycle. So the most interesting stage of the cell, so DNA replication.
Yeah. 24 hours. I mean, what does that mean? Like, is there some, does that include some time in between where you're just sitting dormant, doing nothing, and then 24 hours it takes for it to actively divide it, or is it 24 hours just includes the dormant?
That's a great question. So that includes the whole cycle. And in the cycle, it sits in G1 and does something which kind of lives. It has good time.
So M is just going much faster,
Right. Okay. That's what I was about to say. What's the M phase? The M phase would be very interesting for us because that's the moment when you need to pack your stuff. You need to condense all your genetic information. How long does that take? So M phase. And I left this space because I want to discuss something else. So I'm not. So in the M phase in eukaryotes, that takes of the order of an hour, maybe 2 hours. So to completely compact your chromosomes, that's an hour. But in the extreme case, during the early stages of development, this takes a couple of minutes, and that's absolutely shocking. So this takes of the order of 5 minutes. So you can compact, or like zebrafish can compact all its chromosomes during early stages of development in 5 minutes. In early development, there is no G1, G2. People call this M and S. So it's M and S, M and S, M and S, constant. Replicate and divide.
When do they produce protein?
They don't, because that's the moment when they just live off the maternal proteins.
Oh, you mean really early?
Really early, before zygotic activation, before gastrulation, before everything. I don't know how fast is it in flies. You should know this.
Yeah, I mean in flies it's half an hour before they start producing.
Before they start, and that's 13 cell divisions?
No, they start producing around 8.
Oh, they start producing around 8, and that takes half an hour up to 8.
Yeah.
8 cell divisions take place before.
I can check. Maybe
In-
it's more like an hour, but it's of the order of an hour.
Uh-huh.
A nuclear division takes around 10 minutes.
It depends.
10
It's
minutes.
very variable which
Yeah, so early ones, like first
They take
10
less
cycles.
than, so cell cycle 10 takes 3 minutes total.
So this can be done extremely fast.
I have a question.
Yes.
In these early stages, so is DNA compacted and then it's very
Well,
compacted
the
again?
DNA is compacted and decompacted.
Do the cells do anything else in between?
They need to copy the DNA.
Right.
They need to unpack, copy, pack, unpack. So that's what they call M&S.
Yeah, 1 hour until cell cycle 8.
Okay, so that's less than 10 minutes per cell division. So okay, so this is in the case of like super fast, like less than 10 minutes, few minutes per cell cycle, complete cell cycle. So, but that's in the case when presumably both S and M are done in the overdrive mode, and when there is no G1 and G2 and the proteins are not being produced, so there is everything else is shut down. But they do change during this process. Actually, it's actually quite striking. Okay, so bacteria divide in 20 minutes. Actually, it's an interesting. Well, it's an interesting question. How can bacteria copy its DNA in 20 minutes? We'll come back to this later. I don't want to talk about this now. Coming back to the genes, before we talk about time scales and stuff, so genes, how do cells decide which genes to activate, in other words, express, in other words, transcribe at any moment in time? So there are special regions in the genome that are responsible for gene regulation. And again, the whole field of molecular biology is now fascinated, for the last 50 years, is fascinated with gene regulation, or mechanisms of gene regulation. So how genes are being regulated? Typically, if this was a human gene, let's actually start, so that's a gene. Let's start in bacteria or yeast. It's practically the same. So that would be a gene. So a gene needs to be read, and typical notation is that from left to right. So a machinery will come and will read this gene and produce RNA. So how does this machine decide when to come and which gene to read? This is decided based on DNA sequences right upstream of this gene. So this symbol is used in biology to say that's the start of a gene. And right next to the start of the gene, there are sequences that are generally called regulatory sequences. Regulatory sites, I would say. So depending on which so this regulatory sites can be, for example, occupied by some proteins. And when a protein, so that's some regulatory protein that binds here, it may prevent formation of it may prevent molecule that reads DNA from binding there. So this way, this protein, this green protein, would regulate this gene. It would stop or prevent its transcription. And these proteins are generally called transcription factors. Or people abbreviate them as TFs. Another possibility is that such protein would bind and would, on the contrary, help a machine that is about to read the gene to bind there in some way. So they can activate or repress genes. So other proteins, not those that read, but other proteins, depending on where they bind. So they would recognize a specific motif upstream here, a specific sequence, would bind the sequence, and depending on which protein is bound there, it would either stop gene expression or would activate gene expression. Yes?
Okay, so I have a cell, and the cell needs some protein. Okay, so how does it express its need for that particular protein? Like, what does it do? I mean, so in order to send, it has to send some messenger to know, I need to go to this particular region.
Exactly, exactly. So that's the whole textbook in cell biology. But basically, what happens? The cell feels that it needs something. How does it feel it? For example, it gets some very attractive sugar somewhere around, and it says, "Oh, smells good. I need to have enzymes to digest the sugar." And so smells good means that the sugar binds to a protein, say this green one, modifies its shape, and now this protein is capable of binding somewhere and activating a gene that encodes for the enzyme that can digest this sugar. It can be as easy as this. In some cases, it can be like, for example, there are tons of different things that cells sense. Once they sense something, they activate there. It all goes down to expressing the right proteins. But using this now, you can say, okay, that's interesting. That's one wire of a logic. You can build all sorts of logic using these wires. You can create cycles. You can create lots of different things that would function by themselves. But A activates B, B activates A, or A activates B, B suppresses A. You can have like a toggle switch, and all sorts of things just built on this using this logic, using protein, because this protein is also produced by some other gene. What if this magenta gene encodes for a protein that shuts down the green one? So you can build all sorts of logical devices this way. Okay, but mechanistically or molecularly what's happening, the protein needs to bind the regulatory sequence right next to the start of the gene. That's yeast and bacteria. In human, it's more complicated. In human, somehow, and we don't really know how it works, these proteins can bind regions very far from the start of the gene. So that would be the start of the gene. Remember this symbol. And somehow binding of stuff, of some proteins, let's say one protein, to a specific sequence that is located pretty far from the gene. And far here means, actually there is no simple number. So people don't even know how far these regions are. But somewhere of the order of, so I would say the range is from 5,000 bases, to, there might be examples of the order of 1,000,000 bases. Okay, let's give a range. 10 to the 3 to 10 to the 6 base pairs away. 2 hands like fly up. So that's how far these regions can be, and they somehow influence whether the gene is active or not. Yes, let me ask you first. No, sorry, first here.
So it's far apart in bases, but is it actually physically far away?
That's the question that we're going to be talking about for the rest of the MI3 lectures. How is this whole thing folded in space such that this region may actually be close in space? And I don't think we have a clear answer whether it is actually close in space, because as you'll see, everything is incredibly dynamic. But the question is, how can this region transmit information along the genome by either folding or in some other way such that it acts on the gene? Yes, your question. Okay, so we, yes.
So
Let me
you
ask.
said humans, but we would flies have the same thing, but these don't. So what does it start?
You and flies have it to a less extent, not to insult
Have
people.
They have
Yes, they have this. In fact, enhancers were discovered in flies. And they are somewhere in thousands of, tens of thousands of bases away. We know that it's certainly not in yeast. I know an answer that is kind of being not even published. It's not even put together. But we have roughly an answer when it's all started. It started in early metazoans, in early multicellular organisms. At the moment when a particular protein, or at least it coincides with the moment when a particular protein is invented that is critical for this. So that's our work with Francois Spitz. So his lab actually now has data showing that it happened very early in metazoan evolution, that very early multicellular organisms are like yeast. A little bit later, multicellular organisms, they're more like human mouse.
And are they always on the same chromosome?
They're typically on the same chromosome. So there is a myth that this, okay, in flies it might be different, okay? In flies,
In flies, the known ones are on the same chromosome.
okay, so let me say first about mammals. In mammals there is a myth that there's going to be regulation across. There is one known example of this, which is incredibly special. And there are convincing evidence that this is happening across chromosomes, but this is kind of brand new stuff and kind of hard to say. But there are lots of speculation. Textbooks are saying that this is happening across chromosomes. It doesn't. In flies, there is a special mechanism. In flies, if you remember, I drew sisters, and I drew homologs. So in flies, that's an interesting kind of. That's why I say it's important to think about different. In flies, so if you look at your chromosome 1, your maternal chromosome 1 and your paternal chromosome 1 are in two different parts of the nucleus. Can be close, can be far. In flies, that's not the case. In flies, maternal and paternal chromosomes are aligned like that. So regions on one chromosome can touch regions on the other chromosome. And there are examples that this regulatory region lives not only far, but it lives on the other chromosome, that the regulatory region in one chromosome would activate another one, expression on the other one, because they're so aligned. And they are indeed incredibly aligned.
So you're saying they're aligned but slightly shifted?
They're not shifted. They're aligned. They're not that aligned. They're kind of aligned-ish. But they're pretty aligned.
So
They're clearly not shifted.
whether the maternal or paternal gene is transcribed?
Yes, and that's a great point. And this may, in fact, control whether maternal or paternal gene is transcribed. And there is a mysterious phenomenon of allele-specific expression, where some genes are expressed only maternal copies, some genes only paternal. And in some cells, it's even done randomly. Cell decides randomly that for this gene in this particular cell and in all its lineage, it's going to be maternal copy. And then in this cell and it's all its lineage, it's going to be only paternal copy. And it never changes its mind. A good example is olfactory receptor. Olfactory receptors are plenty of them, and each one is expressed in one and only one cell and all its progeny. So every cell of my olfactory system expresses one olfactory receptor out of 2,000 that we have.
Immune receptors are the same.
An immune, yes. Yes, I'll take a few more questions and move on. Yes.
How many transcription factors can there be? So let me formulate the question in another way. Is it true that, like, for humans, number of transcription factors is much larger, and what matters is physical distance? That's why genome is forced to focus on trace structure, because we can pack more.
Yes. Yeah. So those are two questions. Why? So how many transcription factors? Yes, indeed. Humans have more transcription factors per gene than bacteria do. There are amazing scaling relationships between this. Our colleague Eric van Nieuwengen from Basel studies this, and so he he has a series of papers about scaling between number of transcription factors and the number of genes in different kingdoms of life. That's as far as complexity of transcription factors, and that's when I was drawing this. I kind of hesitate to draw one transcription factor. Typically in eukaryotes, there might be several, and only if this combination of transcription factors is bound here, then the gene is activated. So there is a great deal of combinatorial regulation. But why this becomes distal, why these regions are far from the gene, that's not clear, and this happens in evolution at one moment. And there are multicellular organs. There are eukaryotes that don't use this like yeast. There are simple multicellular eukaryotes that are like yeast. And then the rest of the animals and multicellular organisms are like that. There is this amazing event, and now we can roughly date it. So it happened somewhere in very, very, very early evolution of multicellular organisms.
When there's a transcription and you produce RNA, are you just producing a piece of a copy of the DNA that's one gene long, or is it you do the whole thing?
Yeah, okay. So that's one of those many, many exceptions that were kind of firing in my mind. In bacteria, you typically produce several genes that are right next to each other. In eukaryotes, as a rule, you typically express one.
One gene.
Right, because in one thing that you should realize, okay, so that's a good point. In bacteria, if we kind of wrap up this part of the lecture, in bacteria, the genome is very compact. What do I mean? So it's 1,000 times shorter than the human genome, but the total length of genes is comparable. So as a result, in bacteria, genes go one right after another. And they go in both directions. There are lots of reading and writing. There are lots of genetic information that's encoded on the same strand of DNA. Depending on which direction you read, you get a different gene. There are tons of genes. So basically, we can see what fraction we say. So in human, we had 1% covered by coding. In bacteria, what would be this percent? Give me a number. Why wouldn't you put numbers together? What's the percent of the genome that codes? So this is percent coding. So this is human. Human or mouse. Tell me what you have in bacteria. Right. So if this is 3,000 genes times 1,000 base pairs, so what's the number? Okay, 6. Why 60%?
3,000 times 1,000 divided by 5.
So this is 5 divided by. This is 3 divided by 5. Yeah, 60%. Right. So basically, everything is kind of. So there are some gaps between genes, but genes come in very, very dense arrays. In human, on the contrary, have long stretches of the genome, and they're going to be genes again. So in scale, there are going to be some regions with genes, then they're going to be nothing, and some regions with genes, and then nothing, and then so on. So generally, they're going to be regions that are gene dense, and then gene poor, and then gene dense, and then gene poor, gene dense, so on. Yes?
So what percentage of the genome is covered by a regulatory size, and then what are those gene poor areas?
Okay. So these are gene poor, these are gene dense, or gene rich. Let's draw it. So that's a vastly different picture. So what fraction of the genome is in gene poor regions? To the first approximation, you can say half, because genes are kind of clumped together. Where are regulatory? Typically, regulatory regions are somewhere within these gene-dense regions. Again, the scale of things is such that when I say there is a gene-rich region, scale of this can be, say, a megabase, half a megabase. So I'm talking about 10 to the 5, 10 to the 6 base pairs for each such region. So this is enough space for tens of genes and tens of regulatory regions. Regulatory regions are tiny. And the way we discover them, I'll describe when I describe some of the experimental techniques. And then gene-poor regions are gene-poor. They might be, it's actually interesting, so they might be like one gene, one lonely gene, lost. But maybe actually incredibly important somewhere, not talking to anybody. It would have its own regulatory element somewhere.
Yeah, so I guess my question is, in general, in the genome, those regions that is not genes, like what do they do? What are they?
We don't know. We don't know. So yes, like 1.5% of the genome is coding. That's an important number. How can we find out which regions are important and which ones are not?
You knock it out and see what happens.
Good luck. That's actually it's a good question. So there are two limitations. One is ability to cut stuff out. Now it becomes much easier. Now there are many CRISPR scans. In the last year, basically, people started doing massive deletion of stuff.
Everybody knows what a CRISPR scan is?
No, I will describe it in a second. So there is a technique. I don't want to go into detail, so I need to stay at some level otherwise. So there is a technique to remove parts of DNA. You can target an enzyme. You can create a system where you delete this part of the genome. You can cut and paste. Apple C, Apple B. And you can do Apple C, Apple C, you can cut, not copy, you can cut. Apple X, okay. You can cut, you can delete parts in a very specific way. You can say, I will design sequences that are going to be deleted. You design an experiment where you delete stuff, and then you get lots of cells, and different cells, you delete different pieces. The main question is what happens next. You look at them, and they do fine, but you don't know what the physiological effect is, because presumably it's a mouse cell. You say, okay, maybe I need to grow a mouse out of it. That becomes more expensive, and then you grow a mouse, because people have done this for something that I'll tell you about in a second. You grow a mouse, and the mouse is fine. It kind of moves around, eats, lives, dies. You don't know what was this element that you removed, because this element might be used very rarely. It might be used only if this mouse is exposed to some specific infection or to a specific environment and so on.
Wouldn't be a reasonable way of trying to figure out various concentrations of, various molecules inside the cell, various proteins, and see how deletion of
you can guess it from kind of known biochemistry and from stuff like that. You can do that. I'm trying to push you towards a different answer. So in fact, what else can you do other than go and chop genome into pieces? So you have a genome, right? How do you know which parts are more important than others? Yes.
You can tag them and see when they light up.
Exactly. So that's another possibility. You can make a gene. You can modify a gene such that the cell would fluoresce green when this gene is active. You would know when this gene is being used. That's good, but we're not asking a question, which part of the non-coding stuff is being used? And that's really how we don't know how this information is being used. So we need a more abstract approach. Yes? That's what I was wanting. Yes, when it's conserved. For example, so what do you mean? So maybe you can tell us. Exactly. For example, you can compare whether this piece of DNA had changed in evolution or not. And if it didn't, that would indicate that mutations that had happened there were not good for the organism. So nature did fantastic scans for us with lots of different mutations, and it actually probed for something that's relevant. And it took, hundreds of millions of years, and I don't know how many NIH dollars. So nature was just doing this experiment for us. We can just compare the genomes. And then, so if we compare, for example, coding regions of the genome, say between human and mouse, or human and fish, we see that genes are incredibly conserved. And I can show this maybe at the start of the next lecture, just bring the screen down. Genes are incredibly conserved. But it turned out that regions that are conserved are much bigger than 1.5% of the genome. Generally, we're talking about maybe 10% of the genome that's highly conserved between human and mouse. And we have no idea what's there that's being conserved. And that's really puzzling. Yes?
Isn't that just necessary because it's not sufficient? Like, it just happens to be not very often, but it might just be not useful.
If it happened to be not, okay, so the assumption here is that mutations happen randomly everywhere. You can have, okay, strictly speaking, you need to have an underlying model of how mutations happen. But to the first approximation, you can say that all mutations happen uniformly. It's certainly not true. So in some places, there are more mutations happen than in others. And we can take this into account. But then if you have a long stretch of DNA and there are no mutations, so it's like identical between human and fish, zebrafish. Why? It means that mutations were happening, but mutants did not survive. That's the logic. So mutants did not survive. That's why we don't see these mutations in the evolutionary records, in the currently living species. Yes?
Couldn't it be exactly like in the special case where for some reason the DNA is suppressing mutations?
Mutations, right. Is it possible that mutations were never happening there? It's a good question, so it's kind of hard to imagine, but maybe that would be even more interesting case, that somehow mutations never happened there to begin with. And it's actually, for example, for coding DNA, there is a special mechanism that repairs DNA when genes are being transcribed. So mutation happens, if a mutation happens somewhere far from the gene, there is a machinery that fixes mutations, repairs mutations, called the repair machinery. And it does it everywhere. But in the actively transcribed gene, when the gene is transcribed, there is another machinery that comes and makes sure that this gene is particularly mutation-free, this part of the DNA. So yes, repair is stronger, more active in genes. And this is, for example, something that's very clearly seen in cancer genomics. Genes are less mutated, not only because they are needed more, because mutations there would have a strong effect, but also because you have a special machine that repairs genes, protects genes more. Again, nevertheless, at long evolutionary timescales, it's kind of hard to imagine nothing happening in like 10 million years. Or 40 million years.
But the point here is that you cannot sort of like discount the other hypothesis. You cannot basically.
You kind of have to, and so it's a good question. So it's an almost philosophical question for my group, because we always say, oh, maybe there is another biological mechanism. So maybe, but if it's not really known, kind of you need to invoke your biological intuition and what's known and say, no. That has never been seen. It's kind of very unlikely. So you have a price. So it's your Bayesian. So given this data, there are two alternatives. But my Bayesian prior for this alternative is very, very low. That no single mutation happened in this long stretch of DNA over 100 million of years.
No, it could happen. It's just the repair mechanism fixes it.
Yeah, super efficient repair. Yes, it's incredibly important question. Nevertheless, in cancer, when we see some regions not mutating, over short evolutionary timescales, you can ask, maybe this region's never mutated. Then you can look at patients where this region did mutate. And maybe this region did not mutate because it's important for cancer to survive. Or maybe this region never mutated because repair machinery works so well here. And these two things are very hard to disentangle when you're talking about short evolution timescales. Yes, we need to move on.
So if you see a non-conserved region, can you generally assume that that's something that for some reason is not important? Or could you say, like, maybe it's important to me and it's important to the zebrafish, but we both want to do very different things with this region, and so you're both mutating at some reasonable rate?
Right. You can look. That's a great question. When you see a non-conserved region that's different between human and zebrafish, you need to look at other organisms. And then you say, it's different between me and zebrafish. After all, we're different between, they're small, we're big. We assume that we're smarter than that. But then you can look at some other organisms and see how different these regions are. And you can also look at specific patterns of mutations. For example, if you see a gene and you see many mutations that happen in the gene but do not change the protein, that means that somehow the protein needs to be preserved. But you can change DNA without changing the protein because genetic code is degenerate. Okay, so we're talking about like really lots of mutations. So when I say non-conserved, it means you need to look specifically what kind of mutations happen there. But yes, it's possible that regions are actually important for us, important for zebrafish, but we want different things from this genetic part of the genetic information. We just need to look at more species. Yes? Good question.
A question about mutations or non-conserved regions. So actually, is it sometimes also the case that kind of the distance, for instance, is the same, but the sequence kind of changed, so that, for instance, you have a conserved distance between the regulatory regions?
That's a fantastic question. The question is, how do these regions evolve? Are they conserved or not? And are they conserved in sequence or are they conserved in distance? And it's not kind of, so what's known now is that some of these regions are not incredibly conserved. And they might not be conserved for the reason that you just gave. So that, for example, people compare different species of flies, and these regions are not incredibly conserved. Some again, what you need here are positions where these proteins are binding. But as I told you, these regions can be, each of these regions, actually I didn't tell you. So each of these regions is about 10 to the 3 base pairs long. And now people are arguing that they can be even 10 to the 4 base pairs long. These are long stretches of DNA, and they're not completely conserved, but some regions in them can be conserved. But what people notice is that these regions that are conserved, the specific words, imagine there should be specific keywords inserted, embedded in this piece of text. The text is random, but the words should be there. And it doesn't really matter whether the sky is blue or the blue is sky. You can maybe swap these things. And that's what people observed, is that these regions evolve in a very peculiar fashion. But a priori, we wouldn't be able to say that. But you can identify them experimentally and then compare, and you see like swapping of things or copying of words. Blue, blue, blue sky. Blue, blue, sky, blue. Things like this, and maybe that's presumably needed for development of different species of flies. So people compared flies and insects and other insects and so on. Okay, let's wrap it up.
Are the regulatory regions for one gene only?
No, great question. That's what I was about to. No, that's the whole thing.
You have 12 minutes.
Okay, very good, very good. That's roughly what I need. So regulatory regions are somewhere here embedded among genes, as we discussed. This region is 10 to scales. So that's why I started with numbers. It's very important to get a sense of what's happening. So here we're talking 10 to the 5, 10 to the 6 bases. 10 to the 6 is called a megabase. So hundreds of kilobases of the order of a megabase. There are of the order of 10 genes here. And you can have a few regulatory regions there, each a kilobase, each spaced by tens of hundreds of kilobases from a gene. That's all the structure of this region. The most mysterious thing that people observed is that some of these regions, and it's time for me to name them. So these regions are called enhancers. So enhancers are non-specific. Some people call them promiscuous. I'll call them non-specific. So this enhancer, I can insert a reporter gene, a gene that would light up if something activates it. And I can plug this gene in different places in the genome. And I would see that enhancers that are sitting here would activate this gene and would make it light up, would make this gene expressed and would make cell glow green, glow blue in this particular case. But so enhancers seem to activate stuff nearby. How specific they are, we don't quite know. It looks like they can be sufficiently non-specific. The current paradigm in the field is that they just touch stuff. But remember that in bacteria, there are regions right next to the gene that also accumulate some information. In eukaryotes, these regions are still there. And some regulatory proteins bind here. So maybe they activate whatever is ready to fire up. To be ready, you need the right mixture of stuff at the start of the gene. So they might be non-specific.
What's the distinction between enhancers and regulatory sites?
Enhancers are just remote regulatory sites. Let's call them this way. Okay, so now the picture is this. In human, we have this kind of picture of the genome. There are gene-rich regions and there are gene-poor regions. In the gene-rich regions, there are regulatory sequences that act at distance. And the question is, how is this whole thing folded in space? In bacteria, we have a simpler situation. Regulatory regions are near the genes. But again, this thing also needs to be somehow folded in space. Whether the way it's folded in space is important or not, we don't know. But this enhancers or distal regulatory regions, they suggest that maybe it's important how things are folded in space, and maybe that's important for how things work. That's a hypothesis. So the whole field that started booming about 10 years ago kind of operates under the hypothesis that the way the genome is folded is important. We don't know. And we're talking about G1 or G2. So this part of the cell cycle, I should use another color. Okay. This part of the cell cycle, everything other than M is called interface. Interphase, not interface. Interphase. Okay. Everything, when the cell lives, not packs its stuff and divides. That's all about this part. So we know that it's very important how DNA is packed when the cell is about to divide, because it needs to pack its stuff really tightly and then divide. So let's now start talking about the way stuff is folded in space. So now that we spoke about genome in 1D, so that was all genome in 1D part. So now we're talking about genome in 3D. So how is genome folded? Well, first let's get some, okay, I'm looking at the clock. So let's get some estimates of how long is the genome. So what's the length of the human genome? Length of the genome. And again, we're going to do human, yeast, and bacteria. So one base pair is 0.3 nanometers. So how long is the human genome in one cell? Yeah, about 1 meter. Bacteria. I want to look up yeast. I have more accurate number for yeast. Yeast, I gave you 5, 10 to the 7. It's more like 1, 10 to the 7. Sorry. So how long is the yeast genome and bacterial genome? Let's do microns. It's going to be more. Let's do microns. So this is going to be 10 to the 3 microns. Sorry, 10 to the 6 microns. I was thinking about bacteria. Okay, bacteria. About? Yeah, 1.5 millimeters. Again, that's a good sense. So it's like macroscopic, right? Or roughly 1.5, 10 to the 3 microns. And then yeast is, the right number is 10 to the 7. It can be diploid, actually. So yeast can also have a prefactor. Let's be careful. Yeast can also have the same prefactor of 2 or 4, like human, because it's eukaryote, so it's diploid. Yeah, it's twice. Well, should be maybe another factor of 2, maybe 4 times that. Let's say 6 millimeters. Or 6, 10 to the 3 microns. So how big are the nuclei? Human, yeast, bacteria. In bacteria, there is no nucleus, but there is nucleoid, which is a mysterious object, but basically you say the whole bacteria body is kind of filled with DNA. In yeast, there is a nucleus, and in our cells, typically there is a nucleus. So bacteria, how big is a bacteria? More like 1, maybe 2 microns, right? A couple of microns. This is like, say, 0.7. Let's say 1. I don't want to be 1 by 1 micron. Okay, yeast. A few microns, right? So the nucleus, again, we're interested in the nucleus. The nucleus is like easily 3 microns. I think cross-section. Human nucleus, this can be 5 microns in radius, so this would be like 10 microns in cross-section. Again, it might be flat. So this, the thickness of this might be like, say, 5 microns. And the radius is 5 microns. So the question is how to fit 10 to the 6 microns into something of the order of 10 microns, or something of the order of 10 to the 3 microns into 1 micron. You need to fold it and coil it in some way. And so let's take a break at this point, and so then we'll talk about, now we kind of set up the problem. And we can look at what polymer physics would give you as an estimate for how these things are coiled. So taking a break now?
Yes.